Hello, today I will be presenting on the paper The Essential Role of Hippocampal CA1 NMDA receptor dependent synaptic plasticity and spatial memory, and I found this to be an incredibly insightful paper, and I'm very excited to share more about it. I found this to be a really great paper to sum up everything that I had learned in the course, because I found it very interesting, because when I first read the paper, I thought the title sounded interesting, and I liked looking at some of the figures, but I honestly didn't really know what they meant, and I was really unfamiliar with most of the language used in the paper. But as the course went on, and even over the time from when I first wrote my outline to now, I've seen such a greater understanding in what I'm reading, and I think it's really exciting, and I'm really excited to share more about this work. First of all, the authors go into an introduction. That's what they start the paper off with. And they talk about the Hebb rule, which is something we saw in class. And this states that when the presynaptic and the postsynaptic neurons are active simultaneously, their connections become strengthened. And this is really important for memory and synaptic plasticity ultimately. The main receptor that the paper focuses on, they use this acronym NMDARs, and this is a N-methyl D-aspartame receptor, and this is a glutamate receptor and a calcium ion channel that is found in neurons, and it's one of the main ionotropic glutamate receptors, and it plays a very important role in synaptic plasticity, which is the mechanism that is believed to be the basis for memory formation, and they're also involved in some other processes, as we've kind of already talked about. They're also majorly involved in long-term potentiation, which is very important and something that we've covered a lot in class. And this is the notion that there is a persistent strengthening of synapses based on things that you've done before and what you've seen in your past. And it's the idea that these connections between synapses become stronger as you repeat things. So for example, if you're in the video that we watched for the discussion, we saw that they were typing over and over again, and these connections between the synapses and what's firing in your brain is going to be stronger after long-term potentiation is happening. And this is really important in the brain and in learning and in memory ultimately. And the hippocampus has been widely studied in terms of this receptor involvement in synaptic plasticity, and the article goes on to explain a lot of different studies that have been used, and they reference a lot of things that have occurred in the past involving hippocampus and memory formation and all of those things. So now kind of moving on to the core scientific question and the hypothesis. So the main question are that are NMDAR synaptic modifications involved in spatial memory, and I think they very much show their hypothesis in the title, and this is something I didn't really recognize at first because I was having a hard time finding the hypothesis, but then it kind of stuck out to me that the hypothesis could be something that you would see in the title. And so the hippocampal CA1 NMDA receptor dependent synaptic plasticity plays a key role in spatial memory, and we will go on to see that as we look more into the paper itself. So starting with the experimental setup, I found this really interesting, and it's something that I've used a lot in my research lab in terms of looking at knockdown phenotypes, and something that relates a lot to my learning in my biology major. And so they decided to use mice that have a knockdown phenotype, meaning they're not displaying the phenotype, and so you're not going to see mRNA and you're not going to see protein synthesis ultimately creating these receptors in the pyramidal cells of the hippocampal CA1 region. And they wanted to target this area specifically, and they wanted to use a phenotype where you only saw it in that. So they wouldn't have wanted to use something where every single NMDAR is knocked out in the mouse. They only wanted it in that specific region. And so they used a cosmid, and they inserted it into as an exon into certain cells. And so this is really important to understand on how they were able to get the knockdown just in that area. And it was a little confusing at first when I read the paper, because they specified this towards the end, and so I was wondering if they used an RNA interference or something of that sort, but ultimately they get to it, and they used mice that were heterozygous and wild type as their controls, and they verified this through Southern blotting. And so as you can see on the left, something that's heavier is going to move less down the blot because it's harder to move, and you're going to see a larger band if you have more density, versus something that's light, which is the protein on the bottom that doesn't have the NMDAR. It's going to be able to move a lot easier because it's a lot lighter. And so that's how they were able to tell, and they were able to use the different blotting patterns to see whether it was wild type, meaning there's no knockdown at all, heterozygous, which there could be knockdown depending on whether it's like dominant or recessive, but there probably wasn't knockdown if they're using it as a control. And then you ultimately your experimental group is the mice that have this mutation. And in this case, this isn't quite an experiment, it's more to go into the experiments that they're doing, but the dependent variable was the lines on the blot. Furthermore, they then performed microscopy in order to determine any differences in the brain structures in the different mice. And so what they did here is they perfused the brains with PBS, which is a phosphate buffer solution, or phosphate buffered saline, and sucrose, and then they dissected and froze this. Then they performed a cryostat sectioning, and they fixed and stained the different sections that they had just created. And they used two different types of microscopy here. They start with using light microscopy, and with the staining they could see a lot. And they weren't looking for anything too minute. It was pretty big changes that you could clearly see with light microscopy. But then they also used DIC microscopy, which I believe is another type of light microscopy as well. It's just a little bit more specific, and you can see different parts, but it is differential interference contrast, and so that you can use to see more internal structures, so really important. And again, the independent variable throughout this paper is whether the mouse has the NMDAR affecting mutation, but in this case, the dependent variable was what the microscopy image was looking like and what they were noticing when they were doing that microscopy. Now, I feel like when we get into the electrophysiology, we really get into the meat of the paper and what they were trying to look at. And so brains were removed and placed in artificial cerebrospinal fluid, and then they used pipettes to place electrodes in the CA1 region, and then they stimulated. And they used two different stimulation conditions to demonstrate long-term potentiation and then long-term, sorry, I am having a mental blank, but one moment. So they also did LTD in these cases, and they used field excitatory postsynaptic potentials from both the mutant and the wild type mice, and then they also simulated Schaefer collaterals with the high frequency stimulation, and that was to induce the long-term potentiation. And in the wild type mice, they found different results to the mutated mice, and that's how we can see our independent and dependent variables. And they're ultimately measuring those voltage changes because we know we have this voltage gated ion channel, and they wanted to see the difference in postsynaptic current. Ultimately, they took all of these experiments that they were looking at and related them to a Morris water maze test, and this involved the mice picking up blocks and moving throughout this maze, and that was how the scientists related this ultimately to the idea of spatial memory, and they were able to test whether mice were able to repeat this quickly and they were able to do it as fast as the other mice who did not have the mutation. Now to move into results interpretation, we saw a little bit before in the presentation the results of the differences in the genetic situations there, but I wouldn't really consider that as much of a result because it wasn't a clear experiment. It was kind of just to get to this stage of experimentation. They really didn't see many morphological differences in the two brains. In every other brain structure that they looked at the dentate gyrus and a couple other areas. They really saw no difference, and they looked very similar when you saw the actual stainings that they showed in the paper. However, in terms of mRNA expression, you wouldn't see that in the CA1 region because you're not seeing mRNA being produced there. And so that's really important to understand in that case right there. Now, once we go into our electrophysiology and those currents, they found that there was a loss of NMDA receptor-mediated currents and long-term potentiation in this region. And across the other fields, such as here we can see some other fields, they didn't see those, and they look pretty much the same. As you can see in the KA1 region, so in the dark lines, you have those typical peaks that you would see with long-term potentiation, whereas you can see the gray line is staying really flat all the way through. And that's kind of different. I'll try see how these lines are pretty flat, whereas you have these peaks here. That's a pretty big difference that we're noticing, and how we can figure out that you have this complete loss of your synaptic current. Whereas in the other cases, you have your presynaptic function is remained intact, and you also have areas in other hippocampal subfields are remaining intact as well. I didn't touch too much on the results of those behavioral tests, but in the Morris-Water maze, the mutated mice showed severe deficits, and it was very interesting because they performed pretty normally on things that did not involve spatial learning. However, when they were tasked with something that was very specific to spatial learning, that's where they saw, and the scientists used a spatial map. It was a little confusing, but there was poor spatial specificity within that map. Now, I just wanted to go over some of my critiques and overall thoughts of the paper. So first, this raised a question about why there are non-NMDAR pathways and how these are effective. Because if they have these pathways for long-term potentiation, then why do we need NMDARs? And I was just a little confused about this because it seems kind of repetitive, and I wondered why they discussed this in the paper and why it wasn't explained further. Because I think this is something that provokes a lot of thought and something that I hadn't really heard about before. Another thing I noticed is that it's difficult to be certain how well the knockdown affected just the CA1 region, because on the sun blotting, it's difficult to tell exactly where it was, and there's a lot of nearby regions that could easily be affected. So it's interesting because they looked at the dentate gyrus and some other areas in there, but it was interesting that they didn't look at the regions directly surrounding the hippocampus, and they did a little bit, but I think it would be useful to go more in depth. I also think there's a lot of experiments that these scientists could have used, and I think scientists after them have used. This paper was incredibly important and still is over 30 years later, and I think it's just really important to keep looking at and see how we can continue to build on the scientific field using this paper. Furthermore, from a stylistic point, it's difficult to understand the paper when the methods are at the bottom because it left me with a lot of questions. For example, I was wondering if they used RNAi for the knockdown or something else, and ultimately explains what they use at the end, but I think it would have been helpful to have that context going in. Overall, I think the authors have very well justified claims and created an effective scientific argument, but I do think they could have strengthened it by using other spatial memory tests and further expanding on their ideas.